Thank you very much for the invitation. So I know I'm the last one, but I hope you can stay to learn a little bit about the microbiome and the relevance to the early lung cancer. So what is microbiome? Microbiome is, we know, is like microbials inhibiting human body size, like gut, oral cavity, lung, skin. So you can see this figure. We have, and also it a little bit like confused with microbiota, which it's just like community of living microorganisms. But we also care for microbiome. We also care about theater of activities, include the genes, metabolites, and the surrounding environmental conditions, and the host microbial interaction, and like maybe lead to metabolism, immune regulation, inflammation, then the cancer development. So I give you like a brief introduction for gut microbiome. So during the whole life cycles, the microbiome actually is changed. So from neonatal to the old age, you see there's a variation, there's change in the gut microbiome, but they become stable in adulthood. You can see the diet composition, other like nutrition, exercises, or like can have an impact during the adulthood for the microbiome. So in the past few years, I was in MD Anderson Cancer Center, so we complete a few projects on how the diet fiber or probiotics influence gut microbiome. For melanoma immunotherapy response, we found that melanoma patient reporting high fiber consumption had better response to checkpoint inhibitor immunotherapy compared to this patient reporting low fiber diet. And we noticed like every 5 grams increase in daily diet fiber intake correspond like 30% low risk for the progression and death. And most marked benefit was observed for this with patient reporting combination of high fiber consumption and no probiotic supplement use. And we also noticed some like alpha diversity, fiber fermenting, Rhamnococcus Faecalibacterium genus abundance among this like patient with high diet fiber and probiotic use. And we also have a like completed diet intervention. It's called BGON, so moderate prebiotic food source influence inflammation, immune regulating gut microbials and metabolites. So due to time, I will not give like detailed description for this study. And then we also have some like oral microbiome, like research on cancer prevention. So we found that if you have like a higher oral microbiome, alpha diversity have higher all cancer incidence in a cohort of like non-smoking Mexican American women. So yeah, so like I say, we have a lot of like study already to link gut microbiome, link to colorectal cancer, liver cancer, pancreatic cancer. We also like this recent paper on oral microbiome with pancreatic cancer and liver cancer. So, but there's very like less study for lung microbiome is still like imaging evidence on this. So most evidence is associative, mechanistic is still growing. The rationale why, because airways interface with oral derived microbials and lung is not sterile. These biologies may influence field consolidation, chronic inflammation, and pregnant biology. So the key question, can microbial features improve early risk assessment, interpretation of the screen detect lung findings, or prevention treatment target? So just brief introduction for the lung microbiome. Once thought Stora, now recognized as a low bypass ecosystem, is shaped by the aspiration, inhalation, and a host immune tone. And alter like composition seen in the COPD, asthma, and lung cancer, and the microbiome shift in the lung tissue and airways. So there's some evidence, some studies show the oral microbiome linked future like a lung cancer in multiple cohorts. There is a discrimination like microbiome features alone really outperform clinical factors. Multiomics models show the most promise, and there's prognosis therapy context on the gut airway profiles rate to immune checkpoint inhibitor outcomes and peri-operative complications. The bottom line is like this biological plausive and the reproductive signal exists. Prospective pre-specified validation still need to for screen stage use. So the best case context would be like a standardized sampling. We use the saliva sample or oral rinse or induced spectrum with strict contamination control, which is a very challenging for the microbiome sample collection. We use a stable exposure mild antibiotics like PPI, smoking status excluded to reduce confounding. And we use integrated models I mentioned, combine microbiome with age, smoking metrics, radio mix, and inflammation biomarkers. And we need a longitudinal design with baseline follow-up to capture the trajectory rather than one-off snapshots. There is no major guidelines to currently to recommend microbiome testing for lung cancer screening or surveillance. Clinical use still like investigational. Strong endorsement are for the research protocols with harmonized SOP standardized operating procedure data sharing. But microbiome can be a biomarker in early detection, as I mentioned. So we can collect the saliva in clinical use, like in the mobile bus, we can collect the saliva for the lung cancer as well. It may just take 1 minute and stool to show the potential for non-invasive risk stratification. We can combine the microbiome with circulating biomarker, such as circulating tumor DNA, proteomics, radiomics may improve early detection. But be careful, we need to pay attention to the reproducibility and the standardization. So the mechanism for the intervention is majority, like focus on the modulate the immune system. So there's the recent publication to show the details. And then there's a gut lung. We learn a lot about gut liver axis, but there's also like gut lung axis. It's bidirectional communication pathway where gut microbial change impact the lung immune response and vice versa. And there's microbial metabolites, the bacteria produce metabolites that influence the host immunity and then affect tumor development and response to treatment. So there's some strategy for the intervention. One is like FMT. We know like a fecal microbiota transplantation, which we transfer the fecal material from a healthy donor to the recipient and out to the gut microbiome, which can enhance effectiveness of the immunotherapy. And then, as I mentioned in our melanoma project, we use probiotics and prebiotics, which administration of the beneficial bacteria, which is probiotics, or food that promotes the growth, like prebiotics, to influence the gut microbiome and potentially improve treatment outcomes. And then last, nutrition is very important. So my personal research interest is diet intervention. So we can use some we can do high intake of in certain foods such as yogurt has been linked to reduce the lung cancer, which actually is a very interesting paper published in JAMA Oncology. So I'm also interested in some fiber, like beans legume to the cancer for as intervention. Also, the food is medicine is also good strategy, like more holistic diet intervention on the patient. So yeah, this is just example how like the diet, all this cancer therapy integrate with the microbiome modulation and diet intervention to holistic have an impact on the clinical outcome during the cancer therapy. And therapeutic potential, which actually enhanced immunotherapy, improve the chemotherapy effectiveness, like I'm just mentioning it, showing in the last photo, like a figure, and also reducing toxicity, and they also have like an early detection prognosis for the lung cancer. Last, I want to summary for the if you want to do some therapeutic strategy, so there's a microbiota selection, there's diet intervention, and then we can do some omics integration, and it show you like each of the clinical trial design, how do you what kind of study, phase 1, phase 2, phase 3 preclinical studies, and how would you integrate this collection, like a sample biasman collection into your study. Okay, so we do have some challenges like small sample size, heterogeneous methods, contamination, low bypass lung samples, and then standardized pipelines for the multiomics, and also moving from association to intervention. And it takes time for the build intervention for the microbiome. Future direction, we need more like prospective multi-site validation for microbiome, enhanced risk models for clinical utility endpoint. We need a mechanism uncle trials with all these like strategy, and then we need to have a standards for this stand reference for open pipeline for cross-sectional reproducibility. So opportunities like we can integrate microbiome into ongoing early lung cancer cohort. We can combine microbiome with immune, metabolic, genomic profiling. We explore potential as risk predictor on therapy modulators. So yeah, last I want to welcome to collaborate with my microbiome epidemiology lab, and thank you. Thank you.